Sugar is stable. A glucose molecule can sit untouched for years and nothing happens. It won't break down on its own. So your cells face a strange problem. All that energy locked inside glucose is useless unless you can get it out. And to get it out, the cell has to do something that sounds backwards. Before it can take any energy from the sugar, it first has to spend energy. 2 molecules of ATP, just to make the sugar unstable enough to break apart. This is the paradox at the heart of glycolysis. First, where does this happen? Not inside the mitochondria, where most people assume energy is made. Glycolysis happens out in the cytoplasm, the fluid that fills the cell. And it needs no oxygen at all. It runs in completely anaerobic conditions. That's a clue to how old this process is. Because it requires no oxygen and no mitochondria, glycolysis is thought to be one of the most ancient pathways in biology, used by nearly every living thing on Earth for billions of years. Here's the whole process in one view. One 6-carbon glucose gets taken apart into 2 3-carbon molecules of pyruvate across 10 steps run by 10 enzymes in 2 halves. In the first half, the setup, the cell spends 2 ATP to prepare the sugar and split it. Then the molecule divides into 2 3-carbon halves. In the second half, both halves are processed at the same time, and together they yield 4 ATP and 2 NADH. Spend 2, make 4. The net result, a gain of 2 ATP and 2 NADH for every glucose. Step 1. The moment glucose enters the cell, an enzyme called hexokinase spends the first ATP to attach a phosphate group onto it, creating glucose 6-phosphate. And this does something clever. That bulky, negatively charged phosphate acts as an anchor. A phosphorylated sugar can't slip back out through the cell membrane, so glucose is now trapped inside the cell and committed to being broken down. Step 1 is a trap. Step 2 is quiet. An enzyme simply rearranges the molecule. Glucose 6-phosphate becomes fructose 6-phosphate. The exact same atoms, just reshaped. No energy is spent here. It's a small change, but a necessary one. It exposes the right part of the molecule for the critical step that comes next. Step 3 is the most important one. The enzyme phosphofructokinase spends the second ATP to add another phosphate, creating fructose 1,6-bisphosphate. And this is the pathway's main control point, the step the cell speeds up or slows down to regulate the whole process. It's also the point of no return. Once this second phosphate goes on, the cell is fully committed to breaking the sugar down. There's no turning back now. Now, the payoff for all that setup. In step 4, the unstable 6-carbon molecule breaks cleanly into 2 3-carbon molecules. One is ready immediately. The other gets quickly converted so that both become the exact same molecule, glyceraldehyde 3-phosphate, or G3P. And this is the rule to remember. From this point forward, every single step happens twice, once for each 3-carbon half. Everything from here doubles. Step 6 is where we finally start collecting. Up to now, we've only spent. Here, the enzyme oxidizes the 3-carbon molecule. It strips electrons off it. Those electrons are captured by a carrier called NAD+, turning it into NADH, an electron carrier we're storing now to cash in much later. And at the same time, a free phosphate from the surrounding fluid is attached to the molecule, and remarkably, 0 ATP is spent to do it. Remember, this is all happening twice. Step 7. That molecule now transfers a phosphate group directly to ADP, and the first ATP is made. Because this reaction runs twice, Once for each 3 carbon half, the cell generates 2 ATP right here. And that's a milestone. Those 2 ATP exactly recover the 2 the cell spent back at the start. From this moment on, everything the cell makes is pure gain. Steps 8 and 9 set up the finale. First, the phosphate group is moved to a more vulnerable position on the molecule. Then a water molecule is stripped away. The result is a molecule called PEP. And PEP is extraordinarily reactive. It's now desperate to shed its final phosphate. Think of it as a molecule wound tight, holding a phosphate it can barely wait to give away. Step 10. PEP donates its final phosphate to ADP, and the second ATP is generated. Once again, this runs twice. The carbon skeleton left behind settles into its final form, pyruvate. And with that, the cell has now produced more ATP than it spent. 10 steps, 1 split molecule, and a small, reliable profit of energy, every single time glucose comes through. So let's total it up. We started with 1 6-carbon glucose and spent 2 ATP to get going. And the net yield, 2 ATP of immediate, usable energy, 2 NADH, holding electrons in storage for later, and 2 molecules of pyruvate. But here's the honest reality check. This entire 10-step process releases only about 3 to 5% of the total energy locked inside glucose. The vast majority is still there, still trapped inside those 2 pyruvates. So why does this ancient, low-yield pathway still matter so much? Because for some cells, it's everything. Your red blood cells have no mitochondria at all. They can't use oxygen to make energy. Without glycolysis, they would instantly die. They rely on it, 100%, to survive. And it's not just them. Whenever oxygen runs low, high altitude, or the middle of an all-out sprint, glycolysis becomes the emergency lifeline that keeps your tissues alive until the oxygen returns. Ancient, oxygen-free, and universal. But the journey isn't over. Remember, those 2 pyruvate molecules still hold over 90% of the original energy from the glucose. To unlock the rest, they have to leave the cytoplasm and enter the mitochondria, the cell's energy-producing center. And that is where the story continues. In the next episode, the Krebs cycle. If watching sugar come apart, one careful step at a time, made the chemistry inside you feel a little more alive. Stick around. There's so much more to come.